Alright, as promised, it's time for Biochemistry Midterm number 1. So, there's few stuff to go over before we actually begin. This exam that I made is supposed to be really hard. Not to, freak you out or anything. The reason is because I'm trying to replicate and mimic a real biochemistry midterm you would see as an undergraduate student. So if you want those easy questions, go grab your sippy cup and go see Khan Academy. But if you want the real stuff, you're at the right place. I also added some MCAT questions because a lot of people in biochemistry are going to take the MCAT, and biochemistry is a big part of the MCAT. So I decided to include some MCAT style questions as well. Another thing, this is really random, but if you hear a fire truck pass by in the next 5 minutes or so, don't blame me. The fire alarm is going off in the building across from me, so yeah, hopefully it doesn't happen here, but I'm safe. Another thing is that if you find this video helpful, please like and subscribe. Also comment. That really helps me out. But also that if this video does well, I'll probably make a version 2 of midterm number 1. Because I can add so many more questions. But just for time's sake, I limit it up to 10 questions. Let's begin. Question number 1. During glycolysis, fructose 6-phosphate turns into blank using the enzyme blank. What is the product and the enzyme? Support your answer by drawing out all the reactions and products associated and the associated enzyme in this reaction. Okay, so in glycolysis, we're talking about making pyruvate, and there are 10 steps to it. The first thing is glucose turns into glucose 6-phosphate using the enzyme hexokinase. Then from that, glucose 6-phosphate turns into fructose 6-phosphate using isomerase enzyme. And then fructose 6-phosphate turns into what we're talking about here, fructose 1,6-bisphosphate, and uses the enzyme phosphofructokinase. What we do is we take there's the fire truck. Sorry. It takes an ATP molecule, strips the phosphate off. Sorry, give it a second. It's doing its siren thing. Sorry, it's bugging me. Takes the ATP molecule, strips out the phosphate, donates it to the fructose molecule, so it becomes fructose 1,6-bisphosphate. So, here it is right here. Fructose 6-phosphate. We take the ATP, we strip off the phosphate group from it, and it joins carbon number 1. The phosphate group joins to carbon number 1, and we're left with ADP, and the enzyme is phosphofructokinase that does this. B. The enzyme used above is classified as a what? Is it an isomerase, a transferase, a ligase, an oxyreductase, all of the above, none of the above, or I have no idea? Well, hopefully it's not G, because it is one of these answers. A. Is it an isomerase? It is not an isomerase. Because this is not an isomer. Fructose 6-phosphate and fructose 1,6-bisphosphate, the only thing we're adding is a phosphate group. This is not an isomer. What an isomer would be is if it was from glucose 6-phosphate to fructose 6-phosphate. That uses an isomerase. So this is not an isomerase, so it's not A. Is this a transferase? It is a transferase because we are transferring a phosphate group from the ATP onto carbon number 1 here. So this is a transferase. Is this considered also a ligase? This is not a ligase. Ligase is like glue. What ligases do is takes one compound and sticks it together to another compound. That's what a ligase does. And we're not doing this. All we're doing is attaching the phosphate group, but the phosphate group is not a whole molecule itself. If you were attaching fructose 6-phosphate to fructose 1,6-bisphosphate and connect those huge 2 molecules together, that'd probably be a ligase. It's not an oxyreductase because we're not reducing or oxidizing anything here, so nothing regarding electrons. And then it's not E and not F. So it's B, a transferase. 2. In a theoretical experiment, an enzyme is able to work on 3 different compounds. What statement can be said about the enzyme? A. The enzyme is inhibited by compounds 1 and 3. B. The enzyme is allosterically activated by compound 2. C. The enzyme has a high specificity for compound 2. D. The enzyme has a high turnover rate. So the first thing you should do is look at the graph, obviously, and notice that what kind of graph this is. This is a Michaelis-Menten graph. This is not a Lineweaver-Burke plot. Lineweaver-Burke plots are linear. You can clearly see a curve happening here. So this is a Michaelis-Menten. So that's the first thing. Let's go over the answer choices. Let's start with D. D is not correct because turnover rate has nothing to do with Michaelis-Menten. It's a complete separate entity. Nothing to do with turnover rate. So that's immediately wrong. A. The enzyme is inhibited by compounds 1 and 3. Okay, so compounds 1 and 3 are substrates. And we know these are substrates because the bottom x-axis is showing substrate concentration. So these are automatically substrates. Compounds 1 and 3, which are substrates, what A is basically saying is the enzyme is inhibited by compounds 1 and 3. Substrates have no ability to inhibit anything. They're just substrates. Their goal is just turn into products. So they don't inhibit anything. Substrates don't have the capability to do so. Right? So that's why it's wrong. The enzyme is allosterically activated by compound 2. No, it's not, because Michaelis-Menten does not show anything about allosteric activity or anything like that. And plus, once again, compounds are substrates. They don't inhibit anything. They don't inhibit or activate anything, I should say. The only answer is C. The enzyme has a high specificity for compound 2. This is true because you can look at the Km values here. Km value for compound 2 is significantly lower than Km for compound 3 and compound 1, the Km values for those, respectively. So the enzyme has a high specificity for compound 2. Lower Km means higher binding affinity. That means higher specificity. So that's the golden thing you should know is that higher, Km, let's just say lower Km. Lower Km equals higher binding affinity, which equals higher specificity. There we go. 3. Cofactors are critical for maintenance and regulation of metabolic pathways. One of them is unique because it has the ability to accept 1 or 2 electrons. Draw the original molecule and both intermediate molecules and finally the fully reduced form of this electron carrier. So in your mind, you should probably, you should narrow it down to 2 things. It's either NAD or ubiquinone. It's not NAD because NAD does not have 2 intermediate steps or molecules. Only ubiquinone does. Ubiquinone can turn into semiquinone and its relative counterpart here, also known as QH2 with a radical. The fully reduced form is known as ubiquinol. The biggest thing you need to know, the difference is between ubiquinone and ubiquinol. Ubiquinone has 2 double bonds in the ring. Ubiquinol, the fully reduced form, has 3 double bonds in the ring. 1, 2, and 3. This is a key difference. And all you just do, well, number 1, you didn't memorize what this looks like. That's the big thing. But notice that we're just adding radicals and hydrogens to the oxygens. That's basically it. 4. A. The enzyme catalyzed reaction Vmax equals 0.2 moles per second and Km equals 5 millimolar. Assume normal Michaelis-Menten kinetics. What is the reaction rate if the substrate concentration is 10 millimolar? This requires math, unfortunately. I hate math. But yes, we have to do it. So we have to use an equation to solve reaction rate. So we use V equals Vmax times the substrate concentration over Km plus substrate concentration. And solving this will give us the answer. Vmax is given to us, which is 0.2 times the substrate concentration, which is 10, divided by Km, which is 5, plus the substrate concentration, which is 10. This equals 2 over 15, and this is 0.133 moles per second. Yep, and that's the answer. That's the reaction rate, 0.133 moles per second. B. Which enzyme has the highest binding affinity for a substrate given the table? So this obviously is a table for turnover rates. That's what it says, with the given enzyme here. Which enzyme has the highest binding affinity? The answer to this is you cannot tell. This table is a table of turnover rate, not Km. If this is a table about Km, then you can answer this question. But this is turnover rate. Completely different things. Km tells us about binding affinity. Turnover rate tells us how many products are going to make from substrates. Which enzyme will make the least amount of products in a given time period? The answer is lysozyme because it has the lowest turnover rate. Lysozyme. It's pretty easy, right? No. Maybe. Well, let's kick it up in difficulty. You decide to go to McDonald's because you hate your body. Your body's myocytes and hepatocytes are ready to store excess glucose. However, our body must convert the glucose into blank. What is it called? Describe the physiological role the myocytes and hepatocytes have and what differs between them. The first thing you need to know is what hepatocytes and myocytes are. Myocytes are skeletal muscle cells. Hepatocytes are liver cells. What happens is excess glucose is stored as glycogen in the liver and skeletal muscles, but mainly in the liver. Now, when our body needs more glucose, if we're basically energy deficient, it's going to take the glycogen and convert it into glucose. This could happen in the liver and skeletal muscles. So that's what's similar. But what's different is that if the glycogen is stored in the skeletal muscle, it can only be used for skeletal muscle activity like running, walking, jumping, all this kind of stuff. Anything you would use your muscles for. If it's stored in the liver is actually able to transport this glycogen to other parts of the body, wherever it needs it, if it's your brain, your kidneys, your pancreas, wherever it needs it, and then it can convert it to glucose. Okay? So that's the difference. Skeletal muscle is only specific for skeletal muscle stuff. So the glycogen stored in the skeletal muscle can only be used on skeletal muscles. However, the liver can transport the glycogen to different parts. 6. An acyl enzyme intermediate forms during chymotrypsin serine protease protein breakdown, followed by a second nucleophilic attack on the intermediate's carbonyl carbon. Peptide bond is cleaved and enzyme is free. What is the nucleophile in this reaction? Support your answer with the mechanism. Crap. Yeah. So this is probably one of the more difficult questions. It's just remembering the chymotrypsin mechanism. That's it. As long as you remember that, you can get 100% on this question. So water acts as the nucleophile. It attacks the carbonyl carbon. This causes the double bond on basically the bonding from the carbon and the oxygen to disintegrate. The electrons move to the oxygen. It's stabilized by an oxyanion hole. It quickly breaks down, cleaves it off, and the enzyme is free. So that is what is happening here. So first step is the water attacks the carbonyl carbon. The double bond is shredded, basically, and the electrons go up to the oxygen. Oxyanion hole stabilizes for a brief second. Tetrahedral intermediate collapses. Bond breaks. This part joins, right, with the rest of the enzyme. Basically this part goes here, and then this part we just made is free. And yeah, that's it. It's not too bad. It's just from the chymotrypsin mechanism. Alright, what type of inhibitor is this molecule? Molecule X. So, here, actually, I accidentally wrote down the answer. Kind of. So we can see that we have the original reaction right there. The dotted line is after the inhibitor's bind, which is X. Notice that you have a difference of Km and Vmax. From the original reaction, Vmax goes down. We are lowering Vmax. Km also decreases. It goes to the left, right? Because from here to here, Km is decreased. So Km decreases and Vmax decreases. What type of inhibitor is this? This is uncompetitive. In uncompetitive inhibition, what happens is that Km decreases and Vmax decreases. Alright, 8. Although gluconeogenesis is said to be the reverse of glycolysis, there are many key differences. One of the main differences is between the enzyme used in step 1 of glycolysis and the enzyme used in the last step of gluconeogenesis. What are those two enzymes? Prove your answer with reactants, products, and enzymes and intermediates if applicable. First step of glycolysis. Glucose turns into glucose 6-phosphate using the enzyme hexokinase. Gluconeogenesis, glucose 6-phosphate turns into to glucose using the enzyme phosphatase. So, here's glucose. With the help of ATP, we add the phosphate group from the ATP onto carbon number 6. We get ADP, right, as that comes off. In gluconeogenesis, what we're doing is we have glucose 6-phosphate, use the enzyme glucose 6-phosphatase, and phosphatase is basically rip off phosphate groups. With water, it's going to basically shred off the phosphate group here. So there's no more phosphate group on carbon number 6. And that's the difference. Last question. The last step of glycolysis can technically be broken up into two separate reactions. When we put this together, the delta G value, oops, if I to put the delta sign here, delta G is overall negative, very negative. Glycolysis can either move forward to form pyruvate or go backwards to form glucose. Is step 10 a good flux control point? In other words, is it smart to alter the reaction and run it backwards to form glucose instead of pyruvate? The answer is no, it's not smart. Because imagine this. You're running glycolysis. Actually, let me put it in real life terms. Say you're running a marathon and you're almost at the end. You see the finish line. Is it smart to turn around and say, I just quit? It's you know, is it metabolically favored for you to just quit? It isn't, right? You wasted all this energy to finish this marathon, and you just stop and don't want to finish it. What's the point? Same thing about glycolysis, right? You went through 9 other steps, wasted energy, right, to get to this point. What's the point of just turning back around and going back where we came from? It's better to finish the reaction, right, and form pyruvate, because we used all this energy to get to this point, to step 10. And just to turn back around and start from the beginning, why would we want to do that? Right? It's kind of counterproductive, right? Just a waste of energy. So the answer is no. It's not a good flex point. And that is it for the midterm. If you found this video helpful, please like and subscribe. After this, I'm going to talk about the next video on biochemistry. Is going to talk about glycogen. And I think we're on to the Cori cycle, I think. I may be mistaken. But yeah, if you like this video, please like and subscribe. And if you found this really helpful, please comment. And I'll probably make a another version 2 of this midterm and add more questions if you want. So, until next time, later.